Hello and welcome to Progressive Education Society's Modern College of Pharmacy, Nigri, Pune. In this video, we will discuss about in vitro screening methods and their applications for natural products. Specific emphasis on in vitro anti-cancer assay, also known as cytotoxic toxicity assay of the drugs. Before having a look into the anti-cancer assay or activity, we will first know what is exactly cancer in briefly. It is just a condition wherein most of the body cells they begin to divide, and this division is uncontrolled without stopping, and they spread into the surrounding tissue. They can start almost anywhere in the body, in any organ or even in any of the cell or tissue level. These cells range from few millions to few trillions in the particular cell, tissue or organ. There are three kinds of the anti-cancer tumors, like benign tumor, which are localized, and malignant, which can move from one position towards another position. Like, so suppose this is the photograph of benign tumor, whereas this is a metastatic tumor, wherein the tumorous cells they are able to pass through the blood vessels into the adjoining organ. This is called as metastasis. Now, to analyze whether a new drug, whether it is a synthetically derived drug or derived from herbal origin or semi-synthetically derived, these drugs, the anti-cancer potential, also known as cytotoxic potential of these drugs, need to be assayed before need to be determined before uh, conducting the clinical trials or preclinical trials. For that, few of the well-known methods are used. One is called as Sulphorhodamine B assay. This is a dye. Then brine shrimp lethality assay, wherein the shrimps, very small fish-like structures, are killed by the addition of test drugs. Then MTT assay. This is also a dye which helps in identification of the dead cells. Initially, we will look about the principle of this method, sulphorhodamine B assay. Here, the dye, it is able to detect only the viable cells, and the reason for this is it forms a stoichiometric complex with the protein, and only living cells or viable cells are able to produce proteins, whereas the dead cells they cannot produce proteins. It is based upon the ability of this dye to bind with the protein. Generally, basic amino acids are present in almost all the proteins, and the dye specifically binds to the basic amino acids and forms a bright pink color complex. More the living cells, more will be the complex formation, as there is more amount of protein in the living cells. This technique is widely used for analysis of cytotoxic toxic screening of the drugs, or even to test the anti-cancerous drugs, not only on the cancerous cells but also on non-cancerous cells. Here, the binding, as we have seen, is stoichiometric. Stoichiometric means roughly it is in proportion, and the dye that can be separated after killing the cells by addition of DMSO, dimethyl sulfoxide. And once the dye is taken out, the leached dye is separated, and its intensity or the optical density is determined at 500 560 or 565 nanometers. Now we will have a look at the principle in detail. Suppose this is sulphorhodamine B dye, which gives dark pink coloration. If the test tube that has been treated with a sample contains living cells, in that case, these living cells are able to produce proteins. Protein synthesis is on in living cells. So, if suppose our test tube is containing living cells, and if you add the dye to this, the living cells, since they have protein, the dye goes and forms a complex with the proteins. Now, this is the complex. Along with that, the dye also gets accumulated into cytoplasm or can even stick to the cell membrane. The excess dye is to be washed off with acetic acid solution. After taking out the excess dye, now the cells have proportionate amount of dye. Proportionate in sense number of protein and number of dye is proportionate. Here we have seen about 1:1. Next step is to kill the living cells in the test tube. If at all there are any cells, living cells, they are to be killed. Now killing is done, or this lysis is done by addition of DMSO and tris buffer solution and acetic acid solution. Now killing will cause leaching of the cell membrane, so all the dye will come out into the test tube. The dye that comes out is to be measured at 560 nanometer on a optical densitometer, or also called as a plate reader. Now see, here if we get these kind of different test tubes, like we will first discuss about the bright pink color test tube, wherein you have got the intensity of the pink color maximum to its maximum. Maximum pink color will be available in the test tube only if this test tube has maximum number of living cells. Maximum number of living cells will have maximum number of proteins, and therefore maximum number of dye will be bound to the protein even after washing. So once you treat it with DMSO solution, all the dye that has been entrapped into the living cells, it will come out and give this coloration. So this test tube here, which has more absorbance or more color, indicates that the drug what you have added initially is not effective. It is less effective in killing the cells. It has not killed the cells. So whatever cells you have added in the first step are more, they have more viable cells in that. Whereas we'll directly compare it with the extreme left test tube, wherein the intensity of the color is too low. This low color intensity indicates, suppose if this test tube was mixed with doxorubicin solution and then the cells are kept here. Doxorubicin, as we all know, is an anti-cancer drug, is cytotoxic drug. So the cells which are kept at this step, first step here, are having very uh, low number of viable cells. They will be low, or at times it can also be possible that there are no viable cells in this uh, step. So the dye that has been added will go and get entrapped only in the cytoplasm and will can able to stick to the cell membrane. But the moment you wash out, all the dye will go off. So here the proteins will not be able to, since it is a dead cell, it will not be able to form a complex. So maximum dye will be washed out during the washing with acetic acid. So minimum amount of dye will be leached during treatment with DMSO. So this is the test tube treated with suppose doxorubicin or a standard anti-cancer drug. So more uh, the conclusion of this slide is that if you have more number of viable cells, the optical density will be more, the color will be more. Or in other sense, if your test tube has a good or a potential potent anti-cancerous drug or a cytotoxic drug, or if your drug is able to kill more of the cells, in that case the optical density will decrease. Now we will discuss the major steps in the procedure. As such, the procedure is very lengthy. It takes around more than 24 hours or sometimes 2 days to complete this assay procedure protocol. So we will discuss only the important steps in the procedure. This method requires a 96 well ELISA reader or a plate wherein the 96 wells, they are very similar to our test tubes, but they are in very low concentration. The amount is very low. The amount of liquid they can hold is very low. It is maximum up to 1 ml. Now these tests, all the wells are not used. Few can be used. So suppose we in the our we will have a different test tubes, and suppose the first test tube we will call it as control, wherein the control test tube will have only the cells and their nutrient media, so that the cell will be surviving. So now we will be adding these cells into certain wells of the plate. Suppose these are the 8 wells wherein we have added the control or the cells which we have not treated with any anti-cancer drugs. So these are the control wells. Next, our test tube containing a test drug. Test in sense, if you have synthesized a new anti-cancer drug, or if you have synthesized extracted a new drug and you want to test its anti-cancer potential or cytotoxic potential, you will be using it in different concentration. Let us take here an example of, say, suppose X drug in 100 microgram concentration, and we have mixed it with the cells, and the cells are now, or the solution is now spread across these wells. So these lower 8 wells indicate the cells those have been treated with test drug in this concentration, that is 100 microgram. Similarly, we will be increasing the concentration logarithmically. The cells, another set of test tube or set of cells treated with 200 microgram of the same isolated drug. It will be placed here, and 400 microgram test drug. We have increased the dose or concentration of the solution. And in the last, we will be using standard doxorubicin. That means here the cells will be treated with standard doxorubicin solution, and these cells will be fixed in the lowermost area. So this is how fixing of the cell takes place. Once the cells are added, the excess after it is incubated for 24 hours, then the cells settle down, then excess water is taken out or the nutrient media is taken out, then fixing reagents are added. Fixing reagents are very similar to glue, so that they stick the cell to the base of the tubes or base of these wells. Now these are our wells with different test samples, and we have fixed them. After fixing for 24 hours, we have now added, suppose, sulphorhodamine B dye. Now the dye, the concentration and the volume of the dye is constant in all the wells. Next, excess dye is to be washed out. Like the same process we do during staining of our tears. We soak it in a dye and we wash it with water. But here, the washing is to be done with acetic acid. Now, acetic acid will take away the excess dye. Next, the plate that has been washed with acetic acid is to be dried. Now, this is the dry plate, how it appears. Next, in these dry plates or the wells, we need to add DMSO solution so that the cells will get killed or lysed and the dye that is present inside it gets extracted, and it is to be mixed with tris buffer solution so that the dye will now be soluble properly in the tris buffer solution. So you can see here the different shades on upon addition of DMSO. The upper three rows can clearly show high intensity color. Now, this color is to be measured at 560 nanometer with a plate reader. Now, this is plate reader wherein UV rays they pass from upper direction and the detector is present at the lower below the plate. So, simultaneously it can measure the optical density of all the wells. This is the detailed procedure about sulphorodamine B assay. There is no need for us to know the detailed procedure, but if someone of you from you wants to have a detailed protocol, this is it. The first step in that is cell culture. We need to culture the cells, and then the cancer cells are to be cultured, and a specific proportion or specific population of these cells are added into the wells. Suppose in this case 5,000 to 20,000 cells are added per well. This is done by gradual dilution. Then it is to be treated with a compound. Now here in this case, serial dilution of the test compound is prepared and then it is added into the wells. Then cell fixation, a single monolayer will form at the bottom of these wells and it has to be washed with deuterated water and excess water is to be taken out. Then staining with SRB, that is sulphorhodamine B, and then excess dye is also washed out. Next is solubilization. Solubilization of the dye that has been retained by the cells. Next, measurement of the optical density. Once optical density is measured at 560 or 565 nanometer, calculations are done by using this formula. That means percent cytotoxicity of a compound. Let us take an example here of doxorubicin. Percent cytotoxicity of doxorubicin will be equal to optical density of DMSO. DMSO means here the control well minus optical density of doxorubicin divided by optical density of the control into 100. So now this is the way how we determine the cytotoxicity, and we are using 8 different wells for a single test tube. That means we are multiplying it the to by 8 or we are repeating this 8 times, so that 8 times in sense simultaneously, so that minimum errors are there. So this is how anti-cancer potential of drug are assayed by using sulphorhodamine B. Now, after completion of the entire experiment, we should remember that suppose these are the test tubes. The test tube with high intensity pink color is control, that does not have any anti-cancer drug or cytotoxic drug, whereas the test tubes or the wells that have low intensity color or no pink color are more anti-cancerous in nature. That means anti-cancer drugs, they decrease the absorbance of SRB. Anti-cancerous drugs decreases SRB's absorbance. Now another anti-cancer assay, which is very popularly known as MTT. Here the cell viability, that is whether they are living or not, plus whether they are proliferating, increasing in the number, is also determined. This assay is based upon a simple principle of reduction of a dye, that is tetrazolium salt. The yellow tetrazolium salt, that is MTT, is yellow in color, and on reduction by mitochondrial enzymes, that is NADH or NADPH, to um, formazan, that is purple in color. This indicates the viability of the cells. Only viable or active live cells are able to convert the yellow salt into purple formazon, whereas dead cells won't be able to convert it. They will maintain the color of the yellow tetrazolium salt. This resulting intracellular purple formazon is separated from the cell by lysis and then quantified or measured by spectrophotometer. Now we'll have a look at the principle in detail. Suppose this is a living cell. Now we know the living cell is active and has enzymes like NADH or NADPH. So the yellow colored MTT will now be reduced to purple color formazan, and NADH will be converted to NAD. So this is how the living cells convert the yellow color dye to purple dye. Now, in case of dead cells, since the enzymes, mitochondrial enzymes are inactive or are not present, so the yellow color dye remain as such. Therefore, suppose you get after the results, if you after the test, if you get these test tubes, um, the extreme left test tubes, like purple color test tubes, they can be concluded to have low anti-cancer activity because more living cells are present in them. Why more living cells are present is because only living cells are able to convert the yellow color dye into purple formazon. So living cells are more. That means our drug, which was intended to have anti-cancer potential, is unable to kill the cell. So the cells are live. Whereas if you have a look at the yellow color test tubes, here you can see yellow color indicates the cells are dead. The yellow MTT has remained unchanged and given yellow color to the test tube. So our drug, which we have added into the test tube, has higher anti-cancer potential. That means it has killed more cells. That means it is cytotoxic in nature. Dilutions of these um, uh, cells are added with culture media, and then for each of the sample, minimum three wells are prepared. Like for control, three wells; for test, three; test two; test three. And accordingly, and even for standard also, we need to prepare minimum three wells. They are incubated. Then the reagent, that is MTT, is added in each of these wells. Later, the plate uh, is maintained in an incubator for 2 to 4 hours. After incubation, detergent is added into the cell and excess of the dye is taken off. The plate is left with the cover in dark for 2 to 4 hours or at room temperature to allow the cells to stick to the bottom. Then with the help of a diluting agent, the MTT, either yellow or Pomazan, is separated and the absorbance is read at 550 to 600 nanometer. Data interpretation is very similar to sulfur. Thus, the conclusion of MTT assay is that the anti-cancer agents, they kill the living cells, that is, they show their cytotoxic property and maintain the yellow color of MTT. Another anti-cancer assay is brine shrimp lethality assay. Brine, in sense, the water which is more saline, salty than fresh water or river water, but it is less salty than the sea water, is called as brine. Shrimp are the creatures very similar to prawns or even smaller than that. Lethality means here we need to add our isolated compound or extracted compound and see whether these extracts or the isolated compound are able to kill the shrimps. If they are able to kill the shrimps, that means they are able to kill the cells also. That means they are toxic to the cells. Cytotoxicity. It is an in vivo test compared to the earlier two. Now, these are the shrimps which are also known as water monkeys, and the eggs they produce, they are dormant, they can be stored indefinitely, and whenever required, by providing a favorable environment, they can be hatched out into saline water, and naples like larvae can be produced, and the cytotoxic assay is to be performed on the naples. Naples are is a stage in the life cycle of this brine shrimp. We'll have a look at the procedure in detail. The first step in this is hatching of Artemia cyst. Now these Artemia pallens is the organism and it lays thousands of eggs in a single spawn, and these eggs are dormant. They can survive for more than 10-20 years, and they are available in the market in the form of very simple small yeast like granules. And if you want to hatch them, you need to create a favorable environment. Now we'll see what is the exact favorable environment. We need to take a shallow container which should consist of around two unequal chambers. Now these two unequal chambers should be separated with a separator that has fine holes into it. One of the chamber is called as light chamber, wherein we are providing light with the help of a table lamp or an LED bulb, whereas the other end is closed with black colored material in order to prevent the entry of the lights, so that the eggs or the cyst, they hatch out and they travel from the dark direction towards the light. Now here the Artemia now please hatch out and they migrate from the hatching compartment, that is dark, towards the illuminated compartment. Both the chambers should be filled with artificial seawater. That is, the salinity should be maintained to around 3.8% salt solution. The container should be large enough so as to accommodate an air pump, or alternatively you can add the air pump aerator into it to provide oxygen. The molting into larval stage, that is the exact Napoli stage, depends upon the temperature. Generally, the temperature is around 37 degree, plus the quality and quantity of the food. Food required for this brine shrimp are the algal solution. An algae is broken down into solution format, and this solution is given to this compartment. After around 36 hours of incubation at room temperature, the Napolis, they hatch out of the eggs and they get attracted towards the light source. The Napolis, the assay procedure starts from here. That means this is the second part, that is assay. The plant extract or the isolated compound that is to be analyzed for anti-cancer activity is dissolved using a suitable solvent, and a stock solution is prepared. Let us take an example of vincristine sulfate isolated from vinca, and it is dissolved in methanolic solution. But methanol, instead of methanol, um, methanol, ethanol are toxic solvents. They themselves have toxicity. So instead of these alcohols, another solvent should be preferred. Or alternatively, the isolated phytoconstituent can be converted into salt and then can be dissolved into water. The plant extracts in various concentration can be tested. Each vial, that means the test tube, should contain the crude drug plant extract, artificial seawater, and 10 brine shrimps, Napolis. 10 are uh, 10 will not uh, cause crowdy appearance, crowdiness into the test tube. So 10 is sufficient. And the final volume of the test tube should be made up with artificial seawater. The calculation, that means the death of the brine shrimps, is done after 24 hours. That means the test tube is maintained in such a condition for minimum 24 hours, and then the death rate is calculated. Negative control means a test tube that does not contain any cytotoxic material, but the plant extract or the phytoconstituent which is dissolved into a particular solvent, the solvent can be incorporated into, only the solvent can be incorporated into the negative control test. Then data analysis, as far as data analysis is concerned, the brine shrimp assay is to be determined by the toxicity. That means by counting the survived Napolis after 24 hours of exposure to the tested sample. Usually, it should it is preferable that there should not be death in the negative control after 24 hours, or there should be minimum death into the negative control. So again, by applying the similar formula, you can determine the percent cytotoxicity of the compound. And again, standard can also be implemented as a separate set into this protocol.